So chemotherapies and chemo-free therapies, including the best combinations of ibrutinib, the acalabrutinib, the new ibrutinib. Okay, so they cannot cure a great portion of patients. Now we're using CAR T cell therapies. Cell therapies, CAR T cell therapy, what is involved? You take the patient's own blood, you draw them, you take the T cells, you put in the lab, and you use viral to carry your virus that's not harmful, but can carry a gene into the cell, transfer to the T cells, so the gene gets expressed on the top of the T cell. This is the chimeric antigen receptor. It's chimeric to the antigen. The antigen is on the lymphoma cells, okay? Now, you get the chimeric antigen, the T cells will able to recognize this lymphoma cells. But before you do that, you amplify this CAR T cells, chimeric antigen T cells in trillions. Then you put it back. Not only the T cells can recognize the lymphoma cells or leukemia cells, it also by number should outnumber them. This is the best therapy. We are effectively in the cell therapy era. Even the very harsh lymphomas we cannot treat, they zoom through chemotherapy, you give them all the target therapies, they just grow. The monster lymphomas, boom, we can kill it with chemotherapy, cell therapy. For mantle cell lymphoma, this afternoon, I'm going to present the ZUMA-2 study, and it's a huge event, much anticipated, especially by our patients and their families. You see, mantle cell lymphoma is really rare. Large cell lymphoma is very common. So the therapy for CAR T cell therapies for large cell lymphoma were proved 2 years ago. And while the large cell lymphoma benefiting, a lot of patients are in remission, the mantle cell lymphoma, sometimes they don't make it. And I feel the pressure, they feel the pressure, everybody feel the pressure. So this event, this presentation this afternoon, which is very connected with the FDA approval, and is much anticipated event. And actually, we already published the abstract for this at this ASH. The response rate is already over 80%. The CR rate is only 53%. We have 60 patients. In the abstract, we can only describe between 80 patients. The 80 patients already fantastic. Waited until this afternoon when I described the 460 patients. So chemotherapy, chemo-free therapy, target therapy, we are in this era. How about in the future? What if, some of the CAR T cell therapies and all the three modalities cannot cure them? In the future, we are looking into the precision medicine era. And I'm very, very proud that I have been the co-principal investigator on the M.E. Anderson Moonshot project on B-cell lymphoma for the past 5 years. So we are using chemotherapy, target therapy, immunotherapy, cell therapies. CAR T cell therapies is not only cell therapy is not only CAR T. We developed it within the Moonshot called CAR NK cell therapies. Very, very good. But the direction we're heading to is a precision medicine era. That right after, in a few years after CAR T cell therapy, we're going to get precision medicine. What is the precision? We sequence all the tumors. Not only we sequence, but we include the DNA, RNA, proteomics, epigenetics. We also study the biological mechanisms of each patient, why they are resistant to all these therapies. Using all the sequence data, the biological mechanism, best technology, we could decipher and find the Achilles heel of each resistant tumor and a kill that attack the Achilles heel with the precise medicine that's needed. We do not need to use so many. That is the future, called the precision medicine era. It is never a better time to be an academic hematologist. It is never a good time, it's never been a good time to be diagnosed with lymphoma. But this area is growing so much, and it's very, very exciting. I wake up every morning very excited.